Jenna just tweeted, "We just learned about the coolest topic in bio. Hashtag cell membrane permeability. Hashtag only cool kids know." Oh my god, Jenna, the coolest girl that knows the most about cell membrane and permeability, is calling me. Hey Jenna, you want to spit some cool facts about cellular membrane permeability?
Yeah, like, did you know that cells are enclosed in a cell membrane?
Yeah, I totally did. But did you know that the cell membrane has selective permeability properties, allowing certain molecules to pass through it?
Obviously I know that.
Wait. Let me spit some fire. The most common selective permeability membrane is the phospholipid bilayer.
Can we meet up for a study session?
Hey, want to come to the study session with us to learn about cell permeability?
What?
Of course! Let's go!
I hope we get into this study session.
Me too.
This study session is for only people who know about cell permeability. What do you guys know?
I know small and hydrophobic molecules can pass freely through the cell membrane.
Good job, Urry.
Channel proteins function by having hydrophilic channels that certain molecules or atomic ions use as a tunnel through the membrane.
Enjoy the study session.
A fluid mosaic model is a membrane as a fluid structure with a mosaic of various proteins embedded or attached to a bilayer of phospholipids.
Correct. Enjoy your study session.
Integral proteins are not embedded in the lipid bilayer at all.
No, not true. Sorry. Count you. Can you believe she thought integral proteins were not embedded in the membrane at all? She got confused with peripheral proteins. Integral proteins penetrate the hydrophobic interior of the lipid bilayer.
So tonight we're talking about the lipid bilayer.
Oh, someone has a question.
Yes, what is the lipid bilayer and what does it look like?
The lipid bilayer looks like this. Here we have the hydrophilic heads and the hydrophobic tails.
You have a question?
What is hydrophilic and hydrophobic?
Hydrophilic heads means that it has an affinity for water, and hydrophobic tails have a fear of water.
Did you know phospholipids are essential for cells because they make up cell membranes? Their structure provides a classic example of how form fits function at a molecular level.
Wait guys, how do things get in and out of the phospholipid bilayer?
One way is through phagocytosis, which is when a cell engulfs a particle by wrapping its pseudopodia around it.
Another way is through penocytosis, which is when the cell gulps droplets of extracellular fluid into the tiny vesicles.
Unlike exocytosis, a transport vesicle that has budded from the Golgi apparatus moves along microtubules from the cytoskeleton to the plasma membrane. When the vesicle membrane and plasma membrane come into contact, specific proteins rearrange the lipid molecules of the two bilayers so that the two membranes fuse. The contents of the vesicle then spill to the outside of the cell, and the vesicle's membrane becomes part of the plasma membrane.
Oh, I only knew about receptor-mediated endocytosis, which enables the cell to acquire bulk quantities of specific substance. Embedded in the membrane are proteins with specific receptor sites exposed to the extracellular fluid to which specific substances bind.
We all know what diffusion is, right?
Yeah, diffusion is the movement of molecules of any substance so they spread out evenly into the available space.
But did you know that osmosis is the diffusion of water molecules?
And guys, don't forget about active transport, how it requires energy because it goes against the concentration gradient.
And that's the complete opposite of passive transport, which requires no energy because it goes down the concentration gradient.
And we all know that the concentration gradient is the region along which the density of a chemical substances increase or decrease.
Tonicity is so important. It's the ability of a surrounding solution to cause a cell to gain or lose water.
For example, isotonic means no net movement of water across the plasma membrane.
And we can't forget about hypotonic, which is when water will enter the cell faster than it exits, and that could initially make it burst.
Another example is hypertonic, when the cell will lose water, shrivel up, and die.
Have you guys heard of the experiment on Seedless Swampy? Shen Li Lu treated them with 1-methyl cyclopropene stored at 25 degrees Celsius for 10 days and concluded the fruit maintains its firmness and decreased titratable acid and vitamin C contents.